Hey everyone, Dr. Spear here again. Got a new topic for you. Today's topic, or the topic of this lecture, is diffusion, and then I've got a related one on osmosis coming up. These are concepts that you may be familiar with from other biology classes or chemistry or physics classes, but they're so important to biology, and we see them so much, we really need to wrap our head around them. We really need to understand them. And so these topics are relatable to our more recent topics because we've been talking about the cells. And when we talked about the cells, obviously things need to get into the cells and out of the cells. That's just a basic life process. And so we talked about things about, the construction of the cell membrane and how you sometimes have protein channels to let things in and out. And we talked about things like surface area to volume ratios and how they're important for letting things in and out of the cell. And so diffusion and osmosis are also centrally related to getting things into and out of the cell. So to get into the cell or out of the cell, you've got to cross the cell membrane. And so there's basically three broad ways that you can get into and out of the cell: diffusion, mediated transport, or endocytosis. So let's talk about diffusion first. At the molecular level, molecules move around at random. And as they move around at random, they will spread out evenly. And this tendency to spread out evenly as you move at random, that's diffusion. It's nothing more complicated than that. So for example, if you had a bunch of molecules concentrated in a small area, those molecules are just going to move around at random, and they're going to bounce off of each other and bounce off the walls and just move wherever they go. And in a short time, they will be spread out evenly. And that's diffusion. The important thing to remember is that this requires no direct input of energy. The molecules already have energy. That's why they can move around at random. And so you don't have to do anything to spread them out. They will spread themselves out. Now, it's important to realize that there's always energy involved. They had to initially get some energy. So in our example, they started out in a more concentrated state. Well, that's not a natural state. That's a high energy state. So to get in that state, you had to put energy in them and concentrate them. But from then on, through random motion, they will spread themselves out. So there's always an original source of energy, but to get them to diffuse, you don't have to add any extra energy. You don't have to spread them out. They will spread themselves out. So we're talking about stuff crossing membranes. So now let's add a membrane to our example. And so let's say we've got a special kind of membrane called an impermeable membrane, a membrane that doesn't let anything cross it. And the molecules all start out on one side of this membrane. And so here again, we've got the molecules concentrated, but we've got a membrane that is impermeable. And so the molecules will move at random, and they'll spread out on one side of the membrane, but not on the other side. And that's because they can't cross the membrane. Well, this is not a good model for the cell membrane, because the cell membrane is not impermeable, it's semipermeable. So semipermeable means it allows some stuff in and doesn't allow other things in. It's selective. And so let's say that these molecules can cross this semipermeable membrane. So what will happen? Again, we start with the molecules concentrated on one side, and they move around at random, and they will spread themselves out over the entire area. They can cross that membrane. It's almost like that membrane's not there. But what is the net effect here? The net effect is all the molecules started on one side, but then they diffused to the other side. So you got molecules that were able to cross the membrane. And so this is analogous to getting into or out of the cell. They were able to cross the membrane simply by the random movement of molecules. You didn't have to expend any extra energy to get them to cross the membrane. Well, that's diffusion. The molecules were concentrated on one side and they diffused across to the other side. And there's a term that we use a lot. The molecules moved down their concentration gradient. And so when we started, there was a high concentration on one side and a low concentration on the other side. So there's a concentration gradient. There's a gradient from high to low. A gradient is a steady change from one value to another. And so the molecules were on the high concentration side and moved to the low concentration side. And so moving from high concentration to low concentration, that's called moving down your concentration gradient. Okay, so why do we care? Again, this is very important. We're going to see this over and over again. This is a basic way that stuff gets into and out of our cells. So for example, your cell, you have a high concentration of oxygen outside your cells. Why? Because you're constantly taking in oxygen and your blood's carrying it to the cells. But how does that oxygen actually get into the cell where you need it? Well, it's a high concentration outside, low concentration inside. The molecules move at random and they go into the cell. At the same time, your cell's producing carbon dioxide, which you got to get rid of. So there's a high concentration of carbon dioxide inside the cell and a low concentration outside. So if carbon dioxide molecules move at random, they move across the membrane into the blood and you can expel that carbon dioxide. So it just explains many, many processes in biology. And we keep saying it does not require any extra energy. It happens because of the random molecular movement. There's always an initial input of energy. How did that high concentration of oxygen show up outside your cell? Well, you had to spend energy to move the muscles, to expand your lungs, to suck that oxygen in, and to pump the blood. And, you had to do something to get that high concentration. But from that point on, it takes care of itself. So now ask yourselves, once those molecules spread out on both sides of that membrane, is that it? Do they stop moving? Well, of course not. Molecules are always moving. They keep moving, but there's no net movement. And so as these molecules spread out, then they'll move from one side to the other, but an equal amount will move in the other direction. And so you've got an equal movement between both sides. There's still movement, but there's no net movement. That's called a dynamic equilibrium. Dynamic means it's always changing, but it's at an equilibrium. There's no net change. And so the number of molecules moving from left to right equals the number moving from right to left, and they all keep moving. And so there's no net movement after the concentration becomes equal on both sides. At the start of our little experiment, there is a net movement. Here, the concentrations are unequal. You've got a high concentration on one side and a low concentration on the other. So as the molecules start moving around, there's going to be more moving from left to right than there is from right to left. There's going to be a net movement to the right initially. But once those concentrations even out, although there's movement, there's no further net movement. But the molecules keep moving. Now, when we're talking about cells, a lot of times the molecules can't cross the cell membrane directly. They just don't go straight through it. That's the whole point of a membrane, is that it acts like a barrier and controls what goes into and out of the cell. But you'll remember that fluid mosaic model of the cell membrane. There's lots of proteins studded in that membrane that go all the way through. And they make little holes or pores or little channels in the membrane. They give spots for things to move in and out. And so these molecules can diffuse across the membrane. They just have to go through one of those proteins. And so it's just like, you going into a room, or like before the start of class, there's a high concentration of students outside in the hallway, and once the bell rings or once we start, you all don't, you could still move at random and you would spread out, but to get into the room, you got to go through the door. And then once you're through the door, you can spread out even more. It's the same idea. Now, some of these channels are open all the time, but some have gates and are only open some of the time. And so this figure from your book is kind of showing you gated channels. And so it's just a protein that sometimes is open and allows things in and out of the cell, and sometimes is closed. And so what runs the gate, or what opens the gate or closes the gate? Sometimes there's a chemical that's needed to open the gate. And so you've got a chemical that binds to that protein, and then that opens the gate and allows things in. Sometimes it's not a molecule that binds to it, but just a voltage change. There's a change in the voltage or the charge around the gate, and that causes the gate to open or close. So there's different things that can control the gate. So like a voltage sensitive gate, we're going to talk about those a lot when we talk about nerves. The whole function of a nerve is dependent upon this concept of voltage sensitive gates. So at the molecular level, how exactly does this happen, right? How do these proteins make a hole or not make a hole, or make a gate, or open a gate or close a gate? What exactly is going on? Well, remember that these are proteins, and we've made a big deal about saying that the function of proteins is related to their shape, all right? Shape is always important to what their job is. And so for these particular proteins, if they're shaped in one way, that closes the hole. But then if they change their shape, it opens the hole. And so something causes that protein to change shape, which causes it to open or close the gate. That's all that's going on here. Now, another way that we can get holes in the membrane or open some gates is just to push on the membrane and just distort it. And so you can imagine you've got this phospholipid bilayer. It's not a solid entity, and if you just sort of push on it, it opens up pores, and that can let things in or out. So for example, a good example of this is in the lateral line of fish. So again, I'm a fish guy. All my examples have to do with fish. If you ever looked at a fish, you see that most fish have this line that runs down their sides, and they use this to sense nearby vibrations, low frequency vibrations. So they use the lateral line to sense if there are other fish near them. Have you ever seen like a cool National Geographic video where you have this giant school of fish and they're perfectly coordinated, they're all moving in synchrony, and you're like, that's amazing, you got 1,000 fish and they're all choreographed. Well, this is one way they're doing it, is because from this lateral line, they can sense the tail beats of nearby fish, and so they can sense where the fish are around them without even looking. Or perhaps if you're a predator, and then there's some prey back here, you don't have to see it, but you can feel it, and you can turn and get it. That's one use for the lateral line. Another use for the lateral line is if you're in flowing water. And if you sit still and the water's flowing past you, that causes little vibrations that you can sense, and you can sense the speed of the current. So that's what fish use these for. But it doesn't rely so much on protein channels as it is just distortion of the membrane. So here's a sensory hair cell, and you find these in a lot of organisms. You've got them inside your ear. They evolved in fish, and you inherited them from fish ancestors. And what you're seeing here is you've got this one cell, and it's got these stiff hairs on one side, and then it's got a neuron on the other side. So when you push on those hairs, they're stiff, they cause the membrane to distort, which sort of opens up some gaps in it and allows neurotransmitters to leak out. Neurotransmitters are these chemicals that cause a neuron to fire. So that neuron won't fire until it senses, the neurotransmitters bind to it. The neurotransmitters won't be released until something pushes on those hairs. Well, if you've got a vibrating water, that vibrating water pushes on the hair, it distorts the membrane, opens up some pores, lets the transmitters out, the nerve fires. Just another way to make holes in the membrane. Okay, so I've got a couple of just very simple models that I'm going to run for you now, just again so you can picture in your brain this concept of diffusion. So let's take a look at those. Okay, I've got a little model here just to help you think about diffusion. And so when we set this up, what we have are a bunch of dots, and each one of these represents a molecule. And you can see that they're concentrated in the middle. Now each of these is modeled to behave like a molecule in that they just move at random. Whenever I hit go, they'll move at a random speed in a random direction. If they hit another molecule, they bounce off. If they hit the sides, they bounce off. The faster they're going, the harder they bounce. It just is designed to act just like molecules act. And so you can see what happens, and you can imagine what happens. If I hit go and they start moving at random, you can see that pretty quickly they have spread themselves out evenly. And I didn't do anything other than let them move at random. And that's diffusion. And so if we set it up again, now remember, to get molecules in a concentrated state, that does require energy. But from that point on, there's no additional input of energy required. And so when I hit go, pretty quickly those molecules have spread out evenly. And every time I run this, it's different because they're all just moving at random. But that's what molecules do. Now here I've got an example, excuse me, where we've got a membrane in the middle. And so when we concentrate the molecules, they're concentrated on one side of the membrane. And so I've first set this to an impermeable membrane. So the molecules cannot cross the membrane. And when I hit go, those molecules start moving at random. And you see that they spread out, but they only spread out on the side they started on. They can't cross the membrane. And so it's just like the earlier example, but in a more restricted area. And again, when we allow those to move at random, they just spread out evenly. So now we're going to change that membrane to a semipermeable membrane. And this better mimics the cell membrane. And so the molecules, again, are concentrated on one side of the membrane. And as they move randomly, you see they spread out and they spread to the other side. And so what you have is a net movement of molecules from one side of the membrane to the other. And so if we do it again, you can see that these molecules are either concentrated inside the cell or outside the cell. But then if they just move at random, they spread out and there's a net movement across the membrane. And so that's what diffusion is, and that's how you get things to diffuse into and out of the cells. And so once we start, we have a concentration gradient, a high concentration on one side and a low concentration on the other side. And when they start moving at random, more molecules move to the right side than move from the right side to the left side. So there's a net movement to the right side, which you can see. Until the concentration is equal on both sides, and now there's no net movement at this point. The movement doesn't stop, but just as many go from left to right as to go from right to left. But at the start of the simulation, there is a net movement as they diffuse across the membrane. And so that's all diffusion is, and it requires no extra input of energy. All right. So that's probably the most important concept here is diffusion, but we want to build upon that a little bit. Another way you can get across the membrane is mediated transport. And so sometimes molecules need helper proteins to get them across the membrane, to get them into and out of the cell. And so this is very similar to what we just talked about. We just talked about proteins that made holes in the cell, all right? Well, these are similar to that, except these are proteins that actually must grab the molecule and then push it and then move it into the cell, right? The cell just can't go through a hole, or excuse me, the molecule can't just go through a hole. This protein has to grab the molecule and move it into the cell. And this is called facilitated diffusion. And so it's just a similar, it's still based upon diffusion. It still doesn't require any energy, but it's a little bit more complicated than just making a hole in the membrane. And so this becomes important in phenomena when these helper proteins are saturated, when there's so much molecule and only so many of these helper proteins, and they're grabbing and moving as fast as they can. That could limit how fast things can get into or out of the cell. Again, it requires no direct input of energy. But this time, before we had those gated channels, and something would come along and bind it to open the gate, but then it would allow a different molecule in. In this example, the molecule itself opens its own gate. That's only the real difference here. And again, what do I mean by opening its own gate? When the molecule binds to this helper protein, by binding to that protein, that causes the protein to change shape. And when it changes shape, that action of changing shape moves the molecule across the membrane. So again, shape and function are always related in proteins. And so in our earlier example, something else is binding to the protein and opening the gate. And that allows stuff to diffuse through. But in facilitated diffusion, it's the molecule itself that binds to the protein changes shape, and then releases the molecule. So diffusion and facilitated diffusion, we mentioned they require no direct input of energy. So these are called passive transport. They will just happen on their own. Once the conditions are set up properly, the molecules will move on their own. So it's passive, requires no action. But what if we need things to go against their concentration gradient? Things will always move down their concentration gradient without any direct input of energy. But sometimes we need to move things against their concentration gradient. Sometimes we need to move things from a low concentration to a high concentration. That's not going to happen spontaneously. That's not going to happen on its own. That is going to require an input of energy. And so if it requires an input of energy, we call that active transport. And usually we get this energy from an ATP molecule. So we mentioned them last time when we were talking about mitochondria. In the future, when we talk about cell respiration, we'll talk a lot about ATP. Remember that ATP is just like the cell's battery. It's just a way to carry energy from one place to another. And so you digest your food, you put the energy of your food into an ATP, then the ATP goes and it does something like this. The ATP uses that energy to pump things against their concentration gradient. And so in this example, you've got a high concentration on the left and a low concentration on the right. And if left alone, the molecules will diffuse from left to right until their concentrations are equal. But what if we needed them to move from right to left? That's not going to happen spontaneously. We have to actively use energy to pump them against their concentration gradient. And that's what this says here. So here's an example from your book, figure 3.19, showing a very common use of active transport. These are what are known as sodium-potassium pumps. And so these will show up again and again throughout the semester. So we want to kind of understand their concept here. And basically work yourself through this figure so that you understand it. What we're looking at, first off, is a cell membrane, right? Notice the phospholipid bilayer. And then we've got these proteins that are studded in the membrane that go through the membrane completely, go to both sides. And that's your sodium-potassium pump in this example. And so what happens is, step one, 3 sodium ions bind to that protein. And then in step 2, an ATP comes along and transfers energy to that protein. ATP is carrying energy, and so it does this by transferring a phosphate to the protein. These are high energy molecules, and we'll talk about these details later, but the important thing to realize is that the ATP is carrying energy, and it got that energy from, in the mitochondria from your food. Now it transfers that energy to the sodium-potassium pump. When this happens, the 3 sodium ions are now released outside the cell. And at the same time, 2 potassium ions bind to the protein. And then in the final step, those potassium ions are moved into the cell. So we have pumped 3 sodiums out and taken 2 potassiums in. But this would not happen spontaneously. We had to expend energy for this to happen. That's why we had to burn an ATP. Now again, specifically, how does this happen? Why does it work this way? This is protein. And so when the sodiums bind to the protein, it changes its shape a little bit, but not enough. Then the ATP, when it transfers energy to that protein, that energy now is used to radically change the shape of the protein. That protein, in this example, will not change its shape by itself. It requires energy to change its shape. That energy comes from ATP. When that happens, it radically changes its shape, which causes it then to expel the sodium out of the cell. At the same time, it's able to grab, because of this new shape, it's able to grab a couple of potassium ions, and then it snaps back to its original position. But in the process, it releases those potassiums inside the cell. And so again, at the molecular level, it's all about these proteins changing shape. And as they change shape, they're able to do work. And that's what I'm saying here. The energy stored in the ATP molecule is what is used to change the shape of that molecule. You don't always need that. Again, in our earlier example with facilitated diffusion, the protein changed shape, but you didn't need extra energy. The molecule itself had enough energy. And when the molecule itself bound to the protein, that was enough to cause it to radically change shape and pump the molecule across the membrane. Here, you're going against the concentration gradient, so you need to put more energy in to make this happen. If we keep this up, what's going to be the net charge outside of the cell? Well, if you're pumping 3 positive sodium ions out and only taking 2 positive sodium ions in, you're having a net export of positive ions. So the outside of the cell is going to be very positive relative to the inside of the cell. This is going to be really important when we talk about nerve cells. This is how nerve cells work. You set this up so that the outside of the cell is positive and the inside of the cell is negative, and from there you can cause the nerve to fire. But to get into that situation, you have to spend energy, and that energy, this slide shows what that energy goes toward. This is really important in living things. Maybe 10 to 40% of the energy you ingest is just to do this. Think about all the stuff your body does, right? And think about why you get hungry 3 times a day and you're eating. Why do you need so much energy? Because a bunch of that energy is just to run these pumps. Well, why? Because these pumps are critical to nerve function. And where do you have nerves? Your whole body and your brain is nothing but neurons. And so to stay alive, you constantly have to set up this voltage gradient so your neurons can work. And how do you set up that voltage gradient? You use sodium-potassium pumps. So, kind of important. All right, last way to get stuff in is pretty straightforward: endocytosis. Just engulf whatever needs to get into the cell. And so this is, again, a figure from your book. And there's different ways you can engulf things, but the big picture is instead of making holes for things to cross, you just take that fluid membrane and you just engulf whatever you want, and then the membrane just sort of, it's like the blob, it just sort of grabs onto itself, and now whatever you engulf is in a vesicle inside the cell, and you can do whatever you need. Okay, so that's diffusion. Next step, we're going to talk about osmosis, and these are 2 very important concepts that you're going to see over and over again. So let me know if you got any questions. See you in a bit.